For this video, we're going to be talking about viewing systems. In this group is Taylor Mabe, Nakia Harris, Andrea Taylor, Sarah Nash, and Kayla Billups.
Fluoroscopy is an important part of radiology because it shows a moving image rather than a still one. How we are able to see the image is partly due to the different components of the viewing system. There are three main components: television cameras, like the Vidicon camera tube shown here in the top left, charged coupling devices, as shown in the bottom left corner, and television monitors, which consist of LCD, like the one in the top right corner, and plasma, such as the one in the bottom right corner.
TV cameras. The TV cameras just display the fluoroscopic image. It's kind of like our TVs at home or our computer screens on our computer. It uses an older camera tube. It's either pronounced Vidicon or Plumdicon kind of tube. The camera tube is approximately 15 centimeters or 6 inches in length and encloses the electron gun and a photoconductive target assembly. The diameter of the tube is the same size as output phosphor. So the tube is kind of like our X-ray machines. Has a cathode and an anode, except for it has a target assembly. So how it works is the light from the output phosphor arrives at the target assembly by fiber optics or by a lens system. A steady stream of electrons from the electron gun scans the target assembly from left to right and top to bottom. It's called a raster pattern. As this stream bombards the target, anywhere that there is light intensity from the output phosphor, the image there will be an electrical signal leaving the tube. When scanning a brighter or lighter area, like a barium area, the electrical signal leaving the tube is at a higher strength. The darker or lower, kind of like gas or something, light intensities are a lower strength of leaving the electrical signal. So this is a proportional thing. So the brighter or the higher, the higher strength, and then the darker, the lower the strength.
Charged coupling devices are a light-sensitive semiconductor device that generates an electrical charge when stimulated by light, and it can store the electrical charge into a capacitor. What is also known as a CCD may be coupled to an output phosphor by an image intensifier, either with a fiber optic bundle or an optical lens system. The fiber optic bundle contains a very thin optical glass filament. It is very durable and can last a long time, but it does not allow for spot films. To digitize the charge from a CCD, the electrodes between each pixel are charged in a sequence. They move the signal into the row down to where it can be transferred into a capacitor, then sending on as an electronic signal to a television monitor for other systems. The CCD can also be very helpful for the digital fluoro, as it can get rid of some of the problems associated with camera tubes. The CCD does not have any spatial resolution for this and has less noise.
LCD stands for liquid crystal display. In an LCD television monitor, the pixels are switched on or off electronically using liquid crystals to rotate polarized light. Liquid crystal display screens work on the principle of blocking light rather than emitting light. Because of this, LCDs require a backlight. LCDs are made of two pieces of polarized glass, also called the substrate. Between the two pieces is the liquid crystal material. How LCD screens work. Backlight passes through the first substrate. At the same time, electrical currents cause the liquid crystal molecules to align. This allows varying levels of light to pass through the second substrate and creating the colors and images that you see. Are some disadvantages of LCD screens. Black level. LCD screens are not proficient at producing black and very dark grays. They are also not appropriate for use in dimly lit and dark conditions. Contrast. LCD screens have lower contrast due to the poor black level. Cost. The cost of LCD screens are considerably more expensive than the older cathode ray tube monitors. Viewing angle. With LCD monitors, there are restricted viewing angles that affect the brightness, contrast, and color shown. Some of the LCD monitors' advantages are brightness. These monitors show bright images due to high peak intensity. Power consumption. They are very energy efficient. They consume less than 1/3 of the power the cathode ray tube monitors did, and they also produce little heat. Physical aspects. LCDs are thin and compact. They take up 40% less desk space. Sharpness. At the native resolution, the image is perfectly sharp.
Plasma TV monitors. Plasma monitors are very similar to LCD monitors. Instead of liquid crystals, plasma monitors have a thin layer of pixels. Each pixel contains neon and xenon gases lined with a phosphor layer. When electricity flows through pixels, they are stimulated. Pixels show up as red, green, or blue when they're stimulated. The current through the pixels is modulated by electrodes thousands of times per second, controlling the intensity of light produced, making it possible for plasma monitors to produce billions of shades of colors.
To summarize, there are three main parts that are a part of the viewing system of fluoroscopy. The first mentioned were television cameras. There are two main types: the older camera tube, such as the Vidicon or Plumicon, and the newer type, the charge-coupled device or a CCD. The traditional camera tube is a vacuum tube with an electron gun and photoconductive target. The electrons scan the target, which contains the light from the output phosphor, and the light intensity on the target turns into an electrical signal. A charge-coupled device is a series of light-sensitive semiconductor capacitors. Each capacitor is composed of a photosensitive pixel, and between each pixel are electrodes called row gates. When the CCD is stimulated by light, an electrical charge is created and stored in a capacitor. The capacitor sends the electronic signal. Overall, camera tubes are read out by an electronic beam, whereas a CCD is read by the charge in each pixel. A coupling device is when the television camera is coupled to the output phosphor with either a fiber optic bundle or an optical lens system. The fiber optic bundle is more durable, but the optical lens system allows for spot filming. The electrical signals created are sent to the television monitor, and there are two main types used today. The LCD, or liquid crystal display, has several layers that uses the light to determine the shade of gray or color that displays on the monitor. A plasma display is similar to LCD, but instead of liquid crystals, it uses pixels. These pixels contain gas that is ionized when electricity passes through, which then releases ultraviolet radiation, stimulating the phosphor, which produces visible light, and the color produced depends on the phosphor formula. The viewing system is important to fluoroscopy, as that is how the radiology team is able to see the anatomy being imaged. Hopefully this video has helped you gain a better understanding of what goes into being able to view an image taken under fluoro. Thanks for watching.